Okay, then, so let's talk about the second part of the nephron function, which is tubular reabsorption in the proximal convoluted tubule or PCT. So most of the reabsorption, around 85%, actually takes place in the PCT, right? And the lining of this nephron is made up of a single layer of cuboidal epithelial cell, and these cells actually are adapted for the functions in reabsorption. So they have a microvilli, right? This microvilli increases the surface area of the inner lining, which is facing the lumen, right? These cells have tight junctions that hold the adjacent cells together so that the fluid actually cannot pass between the cells, right? So we'll just compare these with the cells of the glomerular capsule, Bowman's capsule as well. So fluid cannot pass between these cells. So all the substances actually have to be reabsorbed and they must go through the cells. So there's a difference. And then these cells have many mitochondria because they have to provide energy for active transport for sodium potassium pumps. And then these cells also have co-transporter proteins in the membrane, which is facing the lumen. So these are specialized cells which have these four features: microvilli, tight junctions that hold the adjacent cells together. They have many mitochondria and they have co-transporter proteins. Okay, so this is the cuboidal epithelial cells of the PCT that you're looking at, right? So this is the part which faces the lumen, is known as the apical part of the cell. It contains many microvilli, which you can see here, right? Then you can see this cell has many mitochondria for active transport, right? Then you can see this layer actually, the one that faces the endothelium of the capillary, this is known as a basal layer, right? Then you can also see these adjacent cells, like this is one cell, this is another cell. These are actually connected by these adjacent cells are joined, right? So they're held together. So everything will actually pass through the cell. Nothing will pass between the cells. So we'll just compare it with the cells of the Bowman's capsule as well, right? So this is the difference that's visible. Okay, then. So see, just compare it. These are the cells of the Bowman's capsule. So these are podocytes. So here you can see, okay, yes, these have these finger-like projections, but there is a distance between these cells. So you can see there's a gap between two cells. There's a gap through which this filtrate was actually passing through, right? So this is the cell epithelial lining of the Bowman's capsule. And see, this is the epithelial lining of the PCT, right? So see, there is no gap. So the procedure is selective reabsorption because around 85% of the glomerulus filtrate is actually reabsorbed back into the blood, but then urea is not reabsorbed, right? And excess mineral ions and urea, they actually stay behind as filtrate, but the rest of the ions like sodium and glucose, amino acids and water, they are actually reabsorbed back. Okay, so the first step in this whole process of selective reabsorption is the reabsorption of sodium. So these epithelial cells of the proximal convoluted tubules, they use two methods. They use active transport and then they use secondary active transport. There are two methods that they use, and then through this, they selectively reabsorb the molecules. So what is active transport? Active transport is when substances are transported across a membrane against the concentration gradient. And for that, we need to have ATP, we need to have proteins, and that's the reason you'll see a lot of mitochondria in that place. So we're just discussing a while back that these epithelial cells, the proximal convoluted tubule, they have an apical side, which is the tubular side. It faces the lumen, and then it has a basal side, which is the capillary side. So just have a look. So this is the epithelium. These are the microvilli. So see, this is the apical side, which actually faces the lumen here, right? So it has many projections for absorption. And this is the basal side, right, which actually faces the capillary. So this whole cuboidal structure that you see here is the epithelial cell of the PCT. And then you can see that these cells, the adjacent cells, are actually connected through these tight junctions. So there's no gap between these cells. Okay, so these sodium potassium pumps you're going to find along the basal side, right? Because we need to transport this sodium through active transport from these epithelial cells into the blood, right? So we have a lot of mitochondria in this place. So these are sodium potassium pumps, right? So what will happen as the sodium moves out of the cell, so the concentration of sodium inside the cell cytoplasm decreases, right? So this is what happens. The sodium is actually actively being transported out of the cell, but here inside the cell, its concentration will decrease. So here you may see sodium is being transported actively out of the cell into the plasma. So what happens because of this, as the sodium leaves the cell, moves into the plasma, concentration of the sodium here decreases. So what happens? There is a more concentration of sodium here in the filtrate, right? So as osmosis, it actually moves inside the cell then, because the concentration here is more. So it diffuses into the cell because now the concentration in the cell is less. This actually happens because of these co-transporters which are found on the apical side of the epithelial cell, right? So along the basal side, we had these sodium potassium pumps here, right? And along the apical side, we have co-transporters. So what these co-transporters do, as the sodium is actually being diffused in, so along with that, glucose or amino acid would also enter inside the cell. So here you can see, so as sodium goes in through the co-transporters, it can actually take in along with it glucose as well as amino acid into the cell. So the glucose and amino acid actually enter the cell at their concentration gradient. So they enter here. So this whole process is actually known as secondary active transport, because what happened, the active transport happened when sodium actually left the cell, right? But because of active transport of sodium out of the cell, the concentration of sodium here decreased. And then as the sodium from the lumen entered the cell, and because of the co-transporters, along with sodium, glucose and amino acids are also able to enter the cell. So this is the reason this process is known as secondary active transport, right? So that it was only possible because of active transport of sodium. So the second part here, as glucose and amino acids were entering it, they did not require any energy there. They did not directly use ATP. They entered because sodium was entering through the co-transporter, so they also entered along with it. And as they are now inside the cell, they can easily diffuse down the concentration gradient into the blood. So just to review, the concentration of sodium ions in the PCT is actually decreased, right? Because as the sodium ions are actively transported out of the PCT cells into the blood in the capillaries, now due to the concentration gradient, sodium ions diffuse down the gradient from the lumen of the PCT into the cells lining the PCT. This is an example of co-transport, because what is happening, as the proteins which transport the sodium ions, they're also carrying along with them glucose as well as amino acid. The glucose, then it comes inside the cell, glucose as well as amino acid, they can diffuse out from the PCT into the bloodstream. So this is how all of the glucose and all of the amino acid actually get absorbed in the proximal convoluted tubule. So most of the absorption is already done here in the proximal convoluted tubule. Okay, so far we were discussing how the solids actually get reabsorbed. So let's now talk about how water is reabsorbed. So since this filtrate contains lots of water from the blood, because you know the hydrostatic pressure of the plasma was really high in the glomerular capillaries. So a lot of water actually filtered out from there. So this contains a lot of water from the blood, and sometimes we need to reabsorb some of that water as well in order to reduce the water loss. So as these solutes, which are sodium, amino acid, glucose, they are pumped into the tubular epithelial cells. Now this means that water potential in the tubular fluid here becomes higher as compared to the epithelial cell in the blood, right? So water potential here is more and water potential here is less. So you know that water follows the passage of the solutes. Down is water potential gradient. So what will happen because of this, water will actually flow inside the cell and then into the blood, because the water potential here was lower because of a lot of number of solutes, and the water potential here was more. So it actually follows the path, and then through the cell, it moves out into the blood and gets reabsorbed. So most of the water reabsorption is already done in the proximal convoluted tubule, right? And then the later part is done in the collecting duct. So for all this selective reabsorption to happen, the role of epithelium of the proximal convoluted tubule is actually very, very important, right? So as we just discussed, that these apical cells, they have these protrusions, right, which are microvilli, which increases surface area. Then you can see the basal layer actually had these sodium potassium pumps there, right? And the cells actually are connected through tight junctions, so nothing can pass between them, right? And the basal side was also enfolded to maximize the transport of cell into the blood. Then here you can see the co-transporters through which, as the sodium was being pumped in, glucose and amino acid were also being pumped in through secondary active transport, right? And then we also saw a lot of mitochondria there, which actually provide the energy for ATP to actively transport sodium out of the cell into the blood. Besides the specialization of the epithelial cell, the proximal tubule itself is also tangled, and this also increases the distance along which the reabsorption can occur, right? So these are the adaptations which actually increase the reabsorption of selected nutrients and as well as water from the PCT into the blood. So at the PCT, around 80 to 85% of the water is reabsorbed according to the water potential gradient without spending energy. So that's actually this water potential, and actually this reabsorption of water is passive transport, right? And this all happens actually as an obligatory renal absorption, right? All the other substances, they get reabsorbed by the PCT, they get actually reabsorbed against the concentration gradient. So that's why the process was active reabsorption. And these following changes take place under active reabsorption. So what happens, all the glucose present in the filtrate is reabsorbed, all the glucose, right? So the ability of reabsorbing substances by the PCT is known as renal threshold value, right? The ability of reabsorbing substances by the PCT is known as renal threshold value. Each substance has its own significant renal threshold value, but if we talk about glucose, it is 180 milligram per 100 ml, which is a very high value, right? So if the blood glucose value exceeds this, the excess amount will not be reabsorbed, right? So the threshold is 180, right? 180 milligram per ml. So if it exceeds this value, the rest will not be reabsorbed. So this time the excess amount will actually be passed down through the urine, and then this condition is known as diabetes mellitus, because normally glucose is never ever present in the urine. Besides glucose, all of the glucose being reabsorbed, around 50% of sodium ions and amino acids are actively reabsorbed by the PCT. Vitamin C is also reabsorbed, chloride ions, and 50% of urea are also reabsorbed passively, right? Then creatinine is actually actively secreted by the cells of the PCT into its lumen, but creatinine and sulfate will not be reabsorbed, right? So what is reabsorbed is sodium, vitamin C, chloride, amino acid, and water. Okay, so as the filtrate leaves the proximal convoluted tubule in the cortex, it now passes into the loop of Henle, which is present in the medulla. So this loop of Henle has three parts. It has a descending limb of loop of Henle that you can see as the proximal convoluted tubule actually goes down. So this part here is a descending limb of loop of Henle. Then this part here is known as the proper loop of Henle, right? And then as it goes up, this is known as a ascending limb of loop of Henle. And then it goes up and joins the distal convoluted tubule, and after that it joins the collecting duct, right? So here in this case, actually a sodium ion gradient has to be maintained, and because of that, further reabsorption of water takes place. So this happens in the medulla, and this happens by the loop of Henle. So here, if you look at the ascending limb of loop of Henle, it's thicker. The walls are actually impermeable to water, but it can actually, sodium and chloride ions can be actively transported out of it. So you'll find a lot of mitochondria here also in this phase, right? But if you look at the descending limb, the walls here are thin, and through that, water can be easily, passively, it can be transported out. So this side is permeable to water, while ascending limb is actually impermeable to water. Okay, so to understand this part better, we always start from the ascending limb. So this sodium and chloride ions are actually being actively transported out of the ascending limb, right? But this side is impermeable to water. So sodium and chloride are actively transported out, but because of that, the concentration of sodium and chloride in the interstitial fluid actually increases a lot. So what happens, because of that, water will actually diffuse out of the descending limb of loop of Henle into the interstitial fluid, because this part here is permeable to water, right? So as water moves out, what happens, as the filtrate goes down, the concentration of sodium chloride here at the bottom of this descending limb of loop of Henle actually increases a lot. So this portion here actually is hypertonic because it contains the most of sodium chloride at this stage, right? And then the sodium and chloride ions, as they go up, they again actually diffuse out of the this ascending limb of loop of Henle. So as the filtrate goes up, so in this part here, it actually is hypotonic because it contains more water as compared to the solids there, because solids have diffused out. So this part here is hypotonic. This part here is hypertonic. So this part here below the dotted line is the medulla. So the tissue in the deeper layers of medulla is actually, it contains a very concentrated solution of sodium chloride. right? And it contains urea as well, right? So, and because of that, as the urine passes down the collecting ducts, because it definitely passes here, so as it passes down the collecting ducts, so water moves out also by osmosis, because already there's a concentration of sodium chloride. So a very strong concentration is there. So water will actually diffuse out of the collecting duct into the capillaries then. So as the blood flows along the vasa recta capillaries, which are these capillaries here, this is the vasa recta, this surrounds the loop of Henle. It receives sodium ions actively and chloride ions passively, right? So this is what happens. So what happens is increases the ion concentration of the blood. Then the blood flows parallel and opposite direction towards the filtrate, which flows downwards along the descending limb. And when these two streams actually come into contact closely, the sodium and chloride ions can enter the filtrate passively, thus increasing its concentration. Due to this, the filtrate becomes hypertonic when it reaches the proper loop of Henle, as we were just discussing. So some amount of water is also reabsorbed in the blood passively at the descending limb of loop of Henle. When the hypertonic glomerular filtrate flows along the ascending limb towards the distal convoluted tubule, it becomes hypotonic. And we were just discussing a while back, it happens because of reabsorption of sodium ions actively, chloride ions passively into the blood. And as this part of the loop of Henle is actually impermeable to water, so no reabsorbing of water will take place in the ascending limb, right? So we just discussed that a while ago. So here, see the whole process, right? So this area becomes hypertonic, this area becomes hypotonic, right? And because of an increased concentration of sodium chloride here, water will actually diffuse out of the descending limb of loop of Henle into the interstitial fluid, and then it can be reabsorbed. Okay, so how is this sodium ion gradient maintained by the loop of Henle? So here in this part, mitochondria in the walls of the cell, they provide energy to actively transport sodium out of the ascending limb of loop of Henle, right? So if you look at number 2, the accumulation of sodium ions in the outside of the nephron in the medulla, it lowers the water potential. See, because you have a lot of solute here now. Therefore, water will actually diffuse out by osmosis into the interstitial fluid in the space in the blood capillaries, and water is reabsorbed into the blood, right? And then at the base of the ascending limb, some sodium ions are transported about by diffusion, as there is now a very dilute solution due to all the water that has actually moved out, right? So here you can see those molalities actually started from 300 here, right? But then it, because of the high concentration, solid concentration here, see, it is the highest here at 1200 here at the proper loop of Henle. And then you see through the ascending limb as a lot of sodium chloride actually is actively being transported. So here you see it's hypotonic, right? So this is a diluted filtrate here right now. It contains less solute, it contains more water. Okay, so a question actually comes in your exam that I suggest how the length of the loop of Henle would differ for a desert animal compared to a human, and they ask you to explain it. So the reason that you are going to give for that is that desert animals actually have a longer loop of Henle. So just think of the reason. We were just discussing a while back that the longer the loop of Henle, what will happen, more sodium ions will actually be transported out, right? So what will happen because of this, water will actually passively diffuse out of the descending limb of loop of Henle. So in this way, more water will be reabsorbed into the plasma, but at the same time, a more concentrated urine actually will be formed here, right? Water will be reabsorbed and concentrated urine will be formed because desert animals, they need to conserve water. So due to all the sodium being actively transported out of the PCT, and then as the filtrate reaches the top of the PCT, this part is actually very dilute, right? So now this filtrate actually has to move into the distal convoluted tubule. This is a distal convoluted tubule, right? So the filtrate, and then actually this section of the medulla, this part here, is very concentrated because it contains a lot of solute here, right? But the part at the top here is actually diluted, right? So because of that, as this filtrate actually passes down the collecting tubule, more water can actually diffuse out because this part is in the medulla now, right? So more water can actually diffuse out of the collecting tubule, right? So what will remain in the collecting duct actually is what will actually form the urine. Okay, so what is happening at the distal convoluted tubule? Sodium ions are reabsorbed actively according to the amount that is present in the blood. Now here at the DCT, this is actually controlled by aldosterone hormone, right? So DCT, this distal convoluted tubule, has an ability to absorb around 15% of the water, right? And this is controlled by antidiuretic hormone. And this we are going to discuss in a while as we discuss osmoregulation. Then chloride ions and bicarbonate ions are also reabsorbed passively according to the blood pH value. And the cells of DCT secrete hydrogen ions, right, and ammonium ions into the filtrate actively, right? So this is the function of distal convoluted tubule. Now, as we talk about the collecting duct, as a glomerular filtrate flows along it, it meets the osmotic pressure gradient which exists from the cortex to the medulla. So as you were discussing in the medulla, the part is very concentrated because of a high amount of sodium chloride. So because of that, about 4.5% of water can also be reabsorbed whenever the hormone antidiuretic hormone is present there. And at the end of it, a very hypertonic or concentrated urine will be formed. So just to summarize, the nephron is made up of renal or Bowman's capsule, proximal convoluted tubule, the loop of Henle, which has a descending limb, a proper limb, and an ascending limb, distal convoluted tubule, and the collecting ducts, right? And these are all surrounded by capillaries. Now, glomerular filtrate is created in the renal capsule. Then glucose and water are reabsorbed into the blood by the PCT, right? All of the glucose and most of the water. Then a sodium ion gradient is actually established here in the medulla by the loop of Henle. And finally, reabsorption of water in the blood occurs in the distal convoluted tubule as well as the collecting ducts. Lastly, let's talk about the tubular secretion. Now, as the glomerular filtrate flows along the tubule, right, so falling substances are actually secreted into the cells by the by the cells of the tubule actually, right? So we have just discussed about what is being reabsorbed, but there are a few things which are actually secreted as well. So what is being secreted from the proximal convoluted tubule? Hydrogen ions, ammonium ions, and creatinine, right? These are secreted by the proximal convoluted tubules, right? The distal convoluted tubule secretes hydrogen ions, potassium ions, ammonium ions, and some drugs as well, right? So these are what is secreted by the PCT as well as the DCT. Now, these two streams of fluid actually, they flow in the opposite direction while associating very closely with each other. Is actually what is known as a counter current system. So this system is actually seen in the kidney, and where it is seen between the vasa recta in the ascending and descending limb of loop of Henle, as you were just looking at it. The function of the system is to maintain an osmotic pressure gradient from the cortex to the medulla. So what happens because of this? Having the two limbs of the loop running side by side like this, with the fluid flowing down in the descending limb of loop of Henle and up in the ascending limb of loop of Henle, this enables the maximum concentration of solutes to be built up both inside and outside the tube, as well as at the bottom of the loop, right? This mechanism is known as counter current multiplier. Okay, so what you see here actually is the microscopic picture of actually the cortical side of the kidney. So as I see, this is a part of the cortex of the kidney basically. So what we are actually looking at, this part here is actually the glomerulus. This part here that you see, right? So this is a glomerulus. So definitely these are capillaries here. So you can see the red part that you see here are the red blood cells, right? This space here is known as the Bowman's space. Okay, or you may call it the Bowman's capsule. So this is the Bowman's capsule. This is its outer wall, right? And this is the white portion that you see is the Bowman's space, right? Now, then what you see here is the cells of the proximal convoluted tubule, right? So as we just discussed, these cells actually the epithelial layers are cuboidal, and they are a little bigger as well. Now, as these are microvilli, so actually here we see a little brush border here. So, but how you can actually differentiate them from the cells of the distal convoluted tubule is that the cells here are actually smaller in size. So see where distal convoluted tubules are, the cells here are smaller, but when you can compare that to the epithelial cells of the proximal convoluted tubule, so these cells are cuboidal and they're bigger in size. So that's how we differentiate them, right? So again, here you see proximal convoluted tubules, right? And this is the cells of the distal convoluted tubules. And this is actually a photomicrograph of the medulla. So here you can see in the collecting duct. So these are the cells lining the collecting duct. And then here you see actually, this is a thin segment of the loop of Henle, and this is a thick segment. So how do we differentiate? So look at the lining. This is a thin segment, right? And this is the thick segment of the loop of Henle, right? So this is whole picture of medulla. So we know that in the medulla, we'll find the loop of Henle and the collecting duct only. While if we talk about the cortex, we'll find the glomerulus, we'll find the Bowman's capsule, we'll find the proximal convoluted tubule as well as the distal convoluted tubule. So these structures are found in the cortex and these structures are found in the medulla. So this is a question from your book. So because you are actually given a question in the paper that you are asked to label the diagram, right? So here they're clearly saying that this is the photomicrograph of a part of a kidney. So we know we are looking at a kidney, right? So this part here, part C that you see right now, is actually the glomerulus, right? This part C. Now, if you look at B here, this part here, right? So you can write it as glomerulus or you can write it as Bowman's capsule. So this is a Bowman's capsule or the Bowman's space, right? Now, if you compare A and D, so look at the cells here. Now, in A, the cells are, the epithelial lining is smaller. But if you look at D, so these are larger in size and they are cuboidal. So this is actually, these are the epithelial cells of proximal convoluted tubule. So look at the size. And these smaller are the epithelial cells of the distal convoluted tubule, right? So now let's have a look at this video where it completely recaps the procedure of how ultrafiltration starts and how this whole reabsorption takes place.
Blood enters the kidney via the renal artery, which divides into smaller arteries, then arterioles. The arterioles get into contact with functional units of the kidney called the nephrons. This is where blood filtration and urine formation take place. The filtered blood then passes through a series of veins and exits the kidney via the renal vein. The urine is collected in collecting ducts and leaves the kidney via the ureters. Each kidney contains over 1 million nephrons. A nephron consists of two major parts, a glomerular capsule, or Bowman's capsule, and a long renal tubule. Renal tubules of several nephrons connect to a common collecting duct. There are 3 steps in the formation of urine: glomerular filtration, tubular reabsorption and secretion, and water conservation. Blood enters the Bowman's capsule via the afferent arteriole, passes through a ball of capillaries called the glomerulus, then leaves via the efferent arteriole. The afferent arteriole is significantly larger than the efferent arteriole, creating a blood flow with a large inlet and small outlet. As a result, the blood hydrostatic pressure in these capillaries is much higher than usual. Hydrostatic and osmotic pressures drive water and solutes from blood plasma through a filtration membrane into the capsular space of the nephron. The filtration membrane acts like a sieve, allowing only small molecules to pass through. These include water, inorganic ions, glucose, amino acids, and various metabolic wastes such as urea and creatinine, and make up the glomerular filtrate. The amount of filtrate produced per minute is called glomerular filtration rate, GFR. GFR is kept at a stable value by several feedback mechanisms within the kidneys, known as renal autoregulation. GFR is also under sympathetic and hormonal control. GFR regulation is generally achieved by constriction or dilation of the afferent arteriole, which causes the glomerular blood pressure to fall or rise, respectively. In a healthy person, the total filtrate volume amounts to between 150 and 180 liters a day. However, only about 1% of this is excreted as urine. The remaining 99% is reabsorbed back to the blood as the filtrate flows through the long renal tubule. This is possible because the efferent arteriole, after exiting the Bowman's capsule, branches out to form a network of so-called peritubular capillaries, which surround the renal tubule. The first part of the tubule, the proximal convoluted tubule, reabsorbs about 2/3 of the filtrate. In this process, water and solutes are driven through the epithelial cells that line the tubule into the extracellular space. They are then taken up by the peritubular capillaries. Sodium reabsorption is most important as it creates osmotic pressure that drives water and electrical gradient that drives negatively charged ions. Sodium levels inside the epithelial cells are kept low thanks to the sodium-potassium pumps that constantly pump sodium ions out into the extracellular space. This creates a concentration gradient that favors sodium diffusion from tubular fluid into the cells. Sodium is absorbed by symport proteins that also bind glucose and some other solutes. Nearly all glucose and amino acids are reabsorbed at this stage. About half of nitrogenous wastes also reabsorb back into the bloodstream. The kidneys reduce the blood levels of metabolic wastes to a safe amount, but do not completely eliminate them. Some of the reabsorption also occurs by the paracellular route through tight junctions between epithelial cells. At the same time, tubular secretion, where additional wastes and other solutes leave the bloodstream to join the tubular fluid, also takes place. The processes of reabsorption and secretion continue in the nephron loop, the loop of Henle, and the distal convoluted tubule. These parts of the tubule also have some other important functions. The main function of the loop of Henle is to create and maintain an osmolarity gradient in the medulla that enables the collecting ducts to concentrate urine at a later stage. The ascending limb of the loop actively pumps sodium out, making the medulla salty. The descending limb is permeable to water, but much less to sodium. As the water exits the tubule by osmosis, the filtrate becomes more and more concentrated as it reaches the bottom. The ascending limb, on the other hand, is permeable to ions, but not water. As a result, the filtrate loses sodium as it goes up and becomes more diluted at the top of the loop. The medulla is in equilibrium with the loop and hence has the same salinity gradient, saltier at the bottom. Reabsorption and secretion in the distal convoluted tubule are under control of various hormones. This is how the kidneys respond to the body's needs and adjust the composition of urine accordingly. The collecting duct receives tubular fluid from several nephrons. The main function of the collecting duct is to concentrate urine and therefore conserve water. This is made possible by the osmolarity gradient generated by the loop of Henle. As it gets saltier deep in the medulla, the filtrate loses water as it flows down the collecting duct. The collecting duct is also under hormonal control, so it can adjust the amount of reabsorbed water according to the body's state of hydration. For example, when the body is dehydrated, more water is reabsorbed back to the blood, and the excreted urine is more concentrated.